A student wanted to investigate the effect of different growing conditions on the rate of photosynthesis in the plant. This is hypothesis statement. After reading this hypothesis statement, we shall paraphrase that in our own words. For example, the effect of the different growing conditions on the photosynthesis rate. So after understanding the hypothesis statement, we shall read the planning and underline the important point, because examiner can design the questions relevant to these important points. For example, dependent and independent variables, and a different variable which remain constant during the investigations, and a different controlled variable during the investigation to whom we shall compare our results, and different potential sources of errors. So during investigations that might possible some point where there are the different potential sources of errors. So we shall identify these following points. Here in the investigations, so the photosynthesis produce oxygen, and this oxygen entrapped in a spongy mesophyll of the leaf, which is helpful to identify the rate of photosynthesis. So in this way, we shall take the two different type of the plant. One type of plant grown in the shaded area, while the second type of the plant, they are grown in the direct sunlight. So we shall cut each leaf into an equal size of the leaf disc and put into syringe. And by pulling and releasing the syringe plunger, we will remove the air from the spongy mesophyll of the leaf. And when all air in the spongy mesophyll of the leaf will be replaced by the water, so then these leaves will settle down in the bottom of the syringe. So when these leaves will settle down in the bottom of the syringe, this will indicate all air has been replaced by water. After that, we shall put these leaves in a separate test tube containing sodium hydrogen carbonate as a source of carbon dioxide. And here in this point, if you observe that, so it is mentioned, the volume of the sodium hydrogen carbonate solution is mentioned by height instead of the volume. So this is our one potential source of error. So in this way, this type of potential source of error we shall underline during the investigations. We're going to see that the investigation statement here, for example, the step number 1, a student selected two plants X and Y, which were of the same species, but they were growing in two different conditions. So in the first step, we need the two different variety to whom we are investigating. For example, the one thing that there are the two independent variables what we have selected. One is the X and Y, two different type of plant, but the constant mean they both belong to the same species. And they are from the two different growing conditions. One is from the shaded area and other is from direct sunlight. Now the step number 2, one leaf was removed from the plant X and we cut the small disc of equal size of 8 millimeter in diameter. So that is the constant variables. Now the student placed the 5 discs from the plant X in the syringe, and by pulling and releasing, we will remove the air from that, and we will put that into the percent of sodium hydrogen carbonate. That is our another constant variables. And after that, we shall place these the plant disc at front of the lamp with the equal distance. So these leaf disc will start to rise, and we shall measure the time, how long they will take to start to rise. And this will help us to indicate the rate of photosynthesis in these leaf discs. Now in this, the investigation, you can see that the concentration of the volume is being mentioned by height instead of volume. So that is what, this is our one potential source of error. The second thing in the step number 5, a distance of 10 centimeter from the edges of the test tube has been mentioned. But when we will observe that this distance is not equal. So in this way, that's also another source of error. And second, time taken for each of the 5 leaf discs start to rise will be measured by only one student. Now, it's not accurately possible for one student to measure or to take 5 different values at the same time. So that is another source of error. So in this way, by reading this, you can pause the video and you can go through from all these different steps and follow the further procedure. So that's our, the student will repeat this point with the leaf disc Y, and then that will proceed to the following point. For example, the distance of all 5 different test tubes from the light source should be 10 centimeter. But when you see that this distance is not equal, for example, that's the point. If you observe carefully from this point, so the distance of the test tube is different. So that is our one potential source of error. So how we can improve that? The distance of each test tube from the light source should be equal. We can put idea. For example, the light source should be in the center and the test tube should around it with the equal distance. Or we can put another idea. For example, we can perform this experiment with each different test tube individually. So that one. Second is another source of error. So for example, 5 leaf discs are present, and these leaf discs will start to rising, might be with the disparity of some time. And in this way, if they are start rising with a different time and one student is measuring their time, it's not possible accurately to measure the same distance or same time or 5 values in the same time. So the time should be staggered, like the student should use this experiment individually with the different test tube, or a student should take the help of the colleagues to measure these 5 days with different timings. So that's our another source of error for us. So we're going to proceed to the next question. After this investigation, examiner will give us the calculated data. This calculated data contains this following values. For example, the plant X leaf disc contain the 5 different values. For example, the first leaf disc took 1 minute and 10 seconds, while the second leaf disc took 1 minute and 5 seconds. And next is a 58 second, and so on. And the same way, this data is present for the plant Y leaf disc. So the question is, prepare the table to record the result shown in the figure 1.4. So whatever the figure is showing us the result, we are supposed to prepare the table for that. So for preparing the table, one thing we should keep in mind, for example, whatever the value is given, all these values should be in same unit. So to convert them into the same unit, we shall select the smallest unit like second, and we will convert these values into the second. For example, 1 minute and 10 seconds is equal to 70 seconds. And in the same way, 1 minute and 5, that will be 65 seconds, and that's already in the second. And in this way, we shall convert these values into the seconds. After conversion into the second, we will draw a table. For drawing the table, so there are, at least we know that there are the 10 different values, 5 for the plant X and 5 for plant Y. So we shall draw a correct number of rows, mean 11. The one row is at the top, and they contain the header, for example, the plant type, the test tube, and the time for leaf disc to start to rising per second. So there is a 1 mark for the heading and 1 mark for correct columns and rows. For example, at least we need the 2 columns and the 2 rows. So at least like accurate rows, like number of rows are supposed to be 11. So that is the most suitable. Second, in the heading, there should be a unit. For example, the time for leaf disk to start rising per second. And the first value, for example, X plant, and with the leaf disk 1, it took 70 seconds. So we'll put this value in the box. In the same way, all these values should be correctly placed in the each row, so they will reward you the 2 marks extra. And this way, there are total 4 marks for these questions. So in this way, we need to be careful. There should not be any unit in the sum boxes. For example, some student put the unit inside of these boxes. So this is the deduction of marks. So here we have completed the 1 question. We're going to proceed to the next question here. In the next question, they calculate the average time taken for the leaf disk from the plant X and the leaf disk from the plant Y to rise. So here the 5 different values are present, and you already have converted them into the second, and we will calculate their average, the time taken. So the average, we will apply the average formula for calculation. For example, the 5 different values, the 70 plus 65 plus 58 plus 86 plus 78. So these are the 5 different values divided by 5, and we will get this value 71 seconds. So that it will be the average time taken for the leaf disk of plant X. So they took 71 seconds. Yes, please. So in this way, what about the second? The average average time taken for the Y leaf disk, that is equal to the 5 different values divided by 5, and we will get this value 229 seconds. So that will secure the 2 marks for you, and we're going to proceed with this value to the next question. Now, on the next question, there is the graph grade is given there, and plot a bar chart on the grade of the average rising time of a leaf disk for the plant X and Y. So in here, there are the 4 marks for that. For these 4 marks, number 1, we are supposed to put the axis. So these axes, for example, along x-axis, this is the independent value, what we have chosen, the plant types. So there are the 2 different plant types we have chosen. The one is the X and one is a Y. And this will give us discontinuous data. And that's our independent variables. What about the dependent variable? This is the result. And this result we will put along y-axis. For example, the average time for the leaf disk start to rising per second. So be careful, there should be proper unit along y-axis. Now, after putting the proper unit, then we shall take the values for which we are going to plot the plant. For example, the plant X took 71 seconds and plant Y 229 seconds. So plant X, so we shall put these values to the accurate point. So that's the bar graph, and there should be an equal distance to another bar graph, and that's the 229 seconds. So there will be two marks for accurately putting these values into the grid, and more than half of the grid should be covered by the drawn values. So in this way, you can secure easily the 4 marks. In the next question, after that, the examiner can ask the question, the type of the different variables. For example, we classify the variables into independent and dependent variables. So the independent variables mean, so what the variable we have chosen, or that is under control of investigator. And these variables do not depend upon other variables. and we can represent along the x-axis of the graph. For example, in this experiment, we can see that the type of the plant, size of the disk, concentration of the sodium hydrogen carbonate solution, and the volume of the solution, temperature, light intensity. If you think that, so these all values are under control of investigator. So the investigator has chosen the different type of the plant. Investigator has chosen the diameter or size of the disk. And investigator, they calculate the sodium hydrogen carbonate solutions. But what about the dependent variables? These dependent variables, the result of experiment, or they depend on the value of independent variables. Of these are represented along y-axis in the graph. For example, the rate of photosynthesis. So, or the time required to start to rising. So how long these lifts will take to start rising is your dependent variables. So these are the values you're supposed to identify in your investigation. For example, suggest why the leaf disk rises when the photosynthesis taken place. So these leaf disks rise when the photosynthesis taken place. What is the reason? For example, when we place them in front of the light, the light source, and they will perform photosynthesis and start to rise. So what is the reason why these leaf disks start to rise? So the reason behind the oxygen produced by photosynthesis is trapped in the air spaces, which reduces the density of the leaf, and the leaf starts to rise toward the surface area, and it makes the disk lighter. So that's any reason you can write, and you will secure 1 mark for that. We're going to move to the second question. Explain why the 5-leaf disc of each leaf type was used. So instead of using 1 leaf disc, why do we use the 5 different leaf discs? Or in the different experiment, why we are using more than 1 different type of variety in that experiment? The reason for that, we calculate average result, so which increase the reliability of the results. For example, it might possible the mistake can be happen, like system errors or human errors might possible. So that's why we calculate more than 1 value and calculate the average of these different values. So this will increase the accuracy or reliability of the results. So that one is the answer we can put. And next question, identify the variable they were measured or the change in this experiment. So what are these different variables, what we measured, and what were the change in this experiment? So the measured variable mean the time taken for a leaf disk to rise to the surface or start to rising. So that is the measured variable, or we can say rate of photosynthesis is our measured variable. While the change variables mean we took the plant from 2 different locations. Some plant were grown in a light area, while some plant were grown in the shaded area. So these are location of the plant or growing conditions of the plant. These are 2 different variables. So this one, that's our secure the 2 marks relevant to that. The next question, state the 2 variables they were kept constant. What are these different variables which we kept constant during this investigations? So mostly like they are the different factors which are affecting on our experiment. So the factor what we are measuring, we will keep them changing. For example, here we took the changing the different type of plant, like the plant grown in shaded, plant grown in light. So these are the changing. What rest of other factor which can affect our result, we keep them the constant. For example, the size of leaf disc, volume of the solution, and the plant species. So these all are constant variables, and we keep them constant. So these are the any 2 points you can write to secure the 2 marks from here. We're going to move to the important questions. What we've been underlining for that any potential source of error in the different past paper examiner usually ask the questions relevant to that in investigations. So there are potential sources of errors in the step number 5, 6, and 7 of the methods on the page number 3 in your original question paper. So in my slide, I don't know about that. So go ahead with that. The page number 3 is a reference of your original past papers. Identify the 2 sources of error in these steps. For each error, suggest the 1 possible improvement regarding that. So step number 5, so the 5 leaf discs from the plant X were removed from the syringe, and each one was placed in a separate test tube with a 2% sodium hydrogen carbonate solution. But here you can see approximately 5 centimeter from the bottom. So instead of giving the volume of the solution, here it is presented height of the solutions. So that is not an accurate value. So this way we can write the improvement regarding that. On second value is a lamp was placed with the distance of 10 centimeter, and we measured in the diagram the distance of all these test tubes were not same. And the lamp was switched on and the timer was started, and the time taken for each of the 5 leaf discs. So to start the rising by 1 student is not accurately possible here. So the error, so measuring height and improvement will be the measure volume. So here it is mentioned this error is measuring height approximately 5 centimeter. So this is not a correct value. So what about an improvement might possible? Instead of measuring the height of the solution, we should measure the volume of the solutions. Second is the error. So leaf disc placed at a different distance from the lamp. So the errors might possible the arrangement. So arrange these with the equal distance of a light source in the center of the circle. For example, we can give the reference like the all different lights, the test tube should be placing around in a circle with an equal distance to the light source. And the third error, so timing multiple leaving a disk. So taking the timing or measure the time rise to the multiple leaf disk, it's not more accurate for 1 student. So in this way, the staggered timing, either we should perform this experiment with a different test tube at the different times, or the student should use the other fellows to measure the time for each different leaf disk. So that one, stagger the timing is an improvement. We're going to move to the next question here. So state the 2 visible differences between the leaf shown in the figure 2.1 and in the figure 2.2 here. So there is in the figure 2.1, one type of the source, the leaf which were grown in the shaded area and the leaf which is grown in the direct sunlight. So from the figure, we can assess that the leaf grown in the shaded area. So obviously there was a less sunlight, less photosynthesis, lean and fragile. So it looked like weak, poor. So in this way, the leaf grown in the direct sunlight, a lot of the supply of light, a lot of food is available, and that's bulky. So in this way, that's our the difference. So the proper difference has been observable is here, and we are supposed to see which is the major difference. For example, if you see that epidermis with the bulky large cells that are present, and spongy mesophyll as a big area is present, mean the large spongy mesophyll is available. So we can put any 2 differences. The sunleaf has the bigger cell, and the sunleaf has thicker palisade mesophyll layer. So here, more important point is that state the 2 visible difference between the leaf shown there. So in this way, you can write either the opposite point, like the opposite point to the leaf in a shade, they are smaller as compared to the sun leaf, or the leaf in the shade, they are the less thick as compared to like palisade mesophyll or spongy mesophyll layer. So these are any two relevant points you can write to secure 2 marks. We can proceed to the next question here. It's a more important point to drawing plant diagram of the leaf. So this is the bifacial leaf, and in this bifacial leaf, you're supposed to draw a plant diagrams. So here there are the 4 marks, and for these 4 marks, we need to justify these 4 points. For example, the outline, it should be 1 mark, sizes or a scale is for 1 mark, and detail 1 is 1 mark, and proportion is for 1. So in this way, you're supposed to draw here. So for example, if there are the 4 different layers of the leaf is present, and these 4 different layers, you're supposed to draw these points. For example, in this mark, according to this marking scheme, first you're supposed to draw the epidermis. Now, this epidermis should be smooth, no individual cell. You're not supposed to use any device by drawing that. So, and there should not be any shading. Second is the palisade mesophyll, and this palisade mesophyll should be broader as compared to the epidermis, but it should be narrower as compared to the spongy mesophyll. And next, you will draw a lower epidermis, and there's a difference between upper and lower epidermis. Why? Because lower epidermis contain stomata that are present inside, and you're supposed to justify the stomatas. For example, here we can keep a small gap in between that. So these are the stomatas that are present. So you're supposed to show that these are stomata with the lower epidermis, and that will create a difference between the upper epidermis and a lower epidermis. Further, the vascular bundle you can put inside. That one is the vascular bundle. In this vascular bundle, so above is the xylem and below is the phloem in bifacial leaves. So this will draw the line, and your line is supposed to be smooth, and size should be bigger than to the more than half. So of the space should be covered by your diagram, and especially you can say your drawn area should be bigger than to the given diagram. And the detailed one, so all 5 or 4 to 5 different layers should be present in that, and proportions should be properly present. We're going to do the practice with that, with this drawing of diagrams. How you can draw a diagram by the pencil in your original papers. If this type of diagram will come, and we shall draw this diagram. For example, the first you're going to draw this diagram of an upper epidermis. So here, this is the 1 layer is an upper epidermis. Second is the spongy mesophyll. That's the palisade mesophyll. Third one is our spongy mesophyll, and we have the third one is our spongy mesophyll, and the fourth one is our lower epidermis. And here we have that the other 2 layers are placed in vascular bundle. Above is the xylem, and below is the phloem. So these are that will become 4 and 5. So these are 5 and 6. So in this way, these are the different layers you can calculate. At least there should be the 4 to 5 layers should be present in your diagram. So we're going to draw this diagram here. So the first is the upper epidermis. By drawing this upper epidermis, you will draw a smooth line by your barehanded. So with the upper epidermis. Second is the layer below, so which will differentiate the upper epidermis. There should be a narrow, and the smooth you will draw by the pencil. And next is the lower line is below here, and this will the palisade mesophyll. This palisade mesophyll should be thicker as compared to the epidermis, but narrower as compared to the palisade and spongy mesophyll. And in the spongy mesophyll below, we have lower epidermis. Now, this lower epidermis contains what? Stomata that are present inside. 1 or 2 stomata you're supposed to justify in that point. This point is more important, like that one. So we cannot not complete that. Why? Because one edge of this this leaf is cut. It's not naturally present, so we will leave it open. Here we are going to draw the open area, and you can see like we justify instead of drawing individual cell, the stomatas. And in this way, this will indicate, and these are the full layer. Now, in this way, in the center, we have vascular bundle, and we can draw vascular bundle in the center. And this vascular bundle, here we have upper is the xylem and below is the phloem. So this will are the complete plant diagrams. Now, in this plant diagrams, you can secure easily the 1 mark when if you do not draw individual cell. There's no overlapping of line. You did not use any scale. You're not supposed to use any scale by drawing these diagrams, and you're not supposed to draw individual cell. For example, some students are drawing like that, individual cell and that. So if you will draw individual cell, or if you will shade this area, for example, here, and if there is the line is going to overlap, for example, this line is going to overlap. So in that way, there will be deduction of 1 mark there. So this way, avoid individual cell by planning by the line diagram and by shading or the overlapping of the lines. And a second, if you will draw, for example, regarding the size, your size should be bigger than to the given area. And a detailed one, for example, when you draw this area, so if you will draw the palisade mesophyll in this point here, so you can see that it is not less than half of that. So in this way, you cannot differentiate. For example, see, there's a proper difference. Like if you see that this distance for palisade mesophyll and spongy mesophyll, there's a big difference. And in this way, what about in your diagram, there is not properly visible. So if you will do like that, and if you will draw this layer here, so in this way, there will be deduction of these marks. There will be detail 1. So, or in this way, proportions, there will be detection for this marks. And in this way, if there is any layer will be less in your diagrams, and there will be deduction of marks. So in this way, always be careful by drawing this type of the diagrams there. So we're going to see that this is the given diagrams. So in the given diagram, these are the given values. We cannot place it here. So in this way, your proper diagrams are like the upper epidermis is there, palisade mesophyll and spongy mesophyll, lower epidermis, stomata are properly leading to the spongy mesophyll, and vascular bundle are properly visible. So in this way, you can easily secure 4 marks for these diagrams. We can proceed to the next question here, the question number 2. So that will be relevant to the measurement, for example, measure the leaf thickness at the line PQ in the figure 2.1 that include the units, and calculate the actual leaf thickness using these equations. So in this way, the first this area has been given between P and Q. So the actual leaf thickness you're supposed to calculate. So the length of the PQ divided by its magnifications. Now this is the length of the PQ you measure by your scale in millimeter. That's coming in 19 millimeter in your past paper's original paper, while below the magnification is given there, that is 130. So the 19 millimeter divided by 130 is equal to 0.15 millimeter. So correct calculations with the correct unit should be present in your answer to secure the 3 marks. We're going to proceed to the next question here. The student measure the length and the thickness of some leaves taken from the different tree. Some part of the tree were in the direct sunlight, and some part of the tree, they were in the shaded. So for example, the leaf grown in the shaded area has been given, and the leaf grown in the direct sunlight has been given. And their average the thickness has been mentioned, and their average length has been mentioned. So we are supposed to calculate the percentage difference in between average leaf length of the leaf grown in the direct sunlight and the leaf grown in a shaded area. And for that, we have the formula. First, we're going to apply the formula for that. So if there is a percentage difference, we're going to calculate. So for the percentage difference, the final value, minus initial value divided by initial value multiplying to the 100, and this will give us percentage difference. For example, here we have the final value of average leaf length is 105 of a leaf in shade. While that one and the leaf in shade, that's the average leaf in direct sunlight is a 70. So in this way, we shall see the difference between these two different values. So for example, that difference is coming 105 minus 70. So, and divided by 70, multiplying to the 100, and this will give us a difference. So first, the difference is 105 minus 70, and then this will give us the value 35. While when we calculate the 35 divided by initial value multiplying to the 100, and we will get this value 50, and that will be our second answer. On the basis of this answer, we can secure the 2 marks, and we can proceed to the next question here. And in this next question, there's a hypothesis statement has been given there. The student was testing the hypothesis. They stated like the leaf grown in shaded area will be longer than to the leaf grown in direct sunlight. So the word longer, so the leaf grown, so in a shaded area will be larger, sorry. So this larger is an adjective word, okay? In the sense larger may be in the sense of length, larger may be in the sense of width, so or thickness. So it's not clear in this hypothesis statement. So in this way, one way we can support this hypothesis, or another way we can reject this hypothesis. Why? Because word larger is not clear. So in this way, that's the point. For example, we can support this hypothesis by this point, mean the shaded leaf is longer in the sense like larger in the sense of longer, like 105 millimeter, while the leaf grown in direct sunlight is less longer, like 70 millimeter. So that's on the one point. We can support this hypothesis like shaded leaf are longer. So that's our supporting this hypothesis like larger with a reference to the longer. So that is the one point. It's a supporting hypothesis. But if we talk about the idea which is not supporting this hypothesis is the shaded leaf might be more thicker. For example, this is thicker. It's been given in this data. The shaded leaf in direct sunlight is 2.27 millimeter. While the leaf grown in the shaded, that's the leaf sunlight, the leaf grown in the sunlight, that's a 2.27 millimeter. While the leaf grown in the shaded, they are 1.53. So there is a difference. So in this way, we can say regarding thickness, we reject this hypothesis. Like this thickness do not support this hypothesis statement. So in this way, 1 mark, you're supposed to write shaded leaves are longer. And another way you can write some leaves are thicker. So that do not support this hypothesis statement. So we have done, we're going to proceed to the next question here regarding the investigations. The B part, before testing the leaf for the presence of starch, the green chlorophyll must be removed. The chlorophyll can be removed by boiling the leaf in the ethanol, and describe how the ethanol can be boiled safely. So you know that the ethanol is an organic compound and it's flammable. So the safety is regarding that the flame should not touch to the ethanol directly. So we can use the water bath, or we can place the test tube containing ethanol in the water beaker. So in this way, the flame will not touch to the ethanol directly. So any 1 point we can write to secure the 1 mark. So here for the second question, suggest why the chlorophyll need to remove. So obviously we are measuring the color blue-black in the presence of the iodine. So the color cannot be measured properly in the presence of the the chlorophyll green color. So to be able to see the color change, so we need to remove the chlorophyll. Now, there are the 5-mark investigations. The student wanted to find out if the starch was present in both leaf grown in direct sunlight and the leaf grown in the shaded area. Plan an investigations to determine if the starch is present in both type of leaf. Now, we have studied the whole hypothesis statement or planning the statement. Now, after studying this planning statement, we shall paraphrase that in our own words. For example, investigate the presence of starch in leaf grown in two different conditions like light and or direct sunlight and the shaded area. So in this way, you will read this all the planning statement, and in this, after reading that, understand and paraphrase in your own words. And after that, you will plan the investigation. For example, we are supposed to take the two different variables, the one variable from the sunlight and the second variable from the shaded, and these three different plant or two different plant belongs to the same species. And after plucking their leaf, we will decolorize them. So we remove the color from that, and we will check with the presence of iodine solutions, so which has the starch, so that is performing photosynthesis. So in this way, we're gonna take one water beaker, and we will boil the different leaves, the one type of leaves and second type of leaf, by putting that into the ethanol containing test tube, and flame should not directly touch to the test tube containing ethanol. So after boiling that, which will remove the color from the green leaves. So these are the green leaves and the ethanol present in the test tube. So the second point, after that, we will take the white tile, and this white tile will help us to identify the color chain with a white transparent background. For example, here we have the petri dish with a transparent bottom, and we will put it on the white tile, and we will put the decolorized leaf in the petri dish. And we apply some drop of iodine solution on that, and this iodine solution will change the color into the blue black, the region which contain the starch. So the region which contain the starch, this will give us a blue black color. So that's the overall the investigations for the 5 marks. And in this investigation, we are supposed to put independent variables. Number one, are we supposed to put that the constant variable? Number two, are we supposed to put that control variables, precautions, and methods? So these everything should be present in our investigations, so then we can easily secure the 5 marks here. So we have some sample planning and investigations, and we can write these planning in the step-by-step. For example, the 3 leaves will be taken from each plant of a same species grown in direct sunlight and in the shaded area. So here we have the independent variables like the 3 leaves. So of the different plant species, different plants belonging to the same species. So, and grown in the different conditions. So after that, the step number 2, boil the leaves in water and then heat in the ethanol containing test tube in a water bath in same duration of times. That's our another constant variables. So it's a constant for both. Flame should not touch to the test tube containing ethanol. So that's our precaution. And after decolorations, rinse the leaf in water and spray on the white tile. And put the same volume of iodine solution in each leaf and observe the color. And the positive test with blue-black color will indicate the presence of starch. So in this way, these are the 5 relevant points we can write, and we can easily secure the 5 marks regarding the planning and investigations. So we have completed all the past papers. Hope you got an idea and it would be more beneficial for you. Thank you. Thank you very much for watching.